Right. Good morning, everybody. It's a pleasure to be here, and I'd like to thank the organisers for inviting me to be part of this panel. As you've heard, I'm from Public Health England, and we are using whole genome sequence analysis to routinely, we're using it routinely to look at, identify and characterise our isolates that we receive both from clinical cases of gastrointestinal disease and also from food isolates that are referred to the laboratory. And we are quite lucky in that we get both clinical isolates and we get the food isolates that are taken either routinely as part of testing and or either as part of investigations. So the organisms you can see that we sequence over on the left-hand side there. And the isolates come in. This is an overview of our process. They come in and we extract DNA from them, put them, check their purity, and carry out the sequencing, which is done by our central sequencing unit. And then once we get the sequencing back, we use that sequence to identify the isolate, and we can also tell usually whether it's mixed or not from the sequence information we receive. We can derive or infer the serotype. We use it to get the MLST type. We discriminate our strains from one another using SNP typing at Public Health England, and this enables us to do cluster detection and to do patient and food isolate matching. So we're using whole genome sequencing for real-time monitoring of a range of GI bacterial pathogens, and it gives us the highest resolution for typing plus phylogenetic information. So it gives us information about how genetically related our isolates are, and this is providing unequivocal evidence that strains are related and from a common ancestor. So it gives us, we've got real-time monitoring of clusters, we've got accurate and robust outbreak definitions. We're detecting more outbreaks than we previously did, and it's giving us lots of advantages, as you can read on the slide, increased case ascertainment and an indication of the source of infection, and potential insights in the geographical location from where either the isolates or the food source came from. So we're getting improved surveillance, outbreak detection and investigation. It targets our epidemiological investigation, so it gives much greater power to those epidemiological investigations. We can look, it gives us information about the genetic relationships of strains over time, so a much greater time frame than previously. We can look at the evolution of emerging pathogens, then that can look at their virulence potential or antimicrobial resistance over time. Improved knowledge about how strains get into the food chain, how they are distributed, better information about transmission pathways, and improved monitoring of public health action, of our public health actions, because the information is just so incredibly powerful. And that's where I'm going to leave my presentation this morning. Thank you.